Okay, now, second, let me say that I am a biochemist, a protein chemist by training, and my group is doing peptide synthesis and bioinformatics for the last 20 years, I have to say. And we started to collaborate with Vittorio, I think, almost 8 years ago when we first synthesized the compound for you. It was a QS signaling molecule, as I understand. I remember it was a long time ago. And then basically we started to work on this communication modeling problem because Vittorio showed us that there are things that can be observed. And this is very important in a modeling study. So now Vittorio actually told me to give a presentation which is non-mathematical and geared towards students that shows the principles of modeling. So why do we do all this? First of all, microbial communities are many, and they are a major form of life. So there are microbial mats under the sea surface, which are bigger than a smaller European country. Okay, and now these things are extremely difficult to study because it is difficult to find measurables on them. You cannot measure the surface. Okay, so and in these cases, engineers, partly like myself, they use modeling because there are things that you simply cannot study directly. For example, if there is a mechanical problem within a nuclear power plant, all they can do is to model it or to shut down immediately. Okay, now with these microbial communities, the situation is somewhat similar because, as I said, those in the open nature cannot be studied very well. So what kind of models do we use? I mean, I tried to make a very superficial classification of the things we do. First of all, we have molecular models. Giordano's talk was a very nice example of using a molecular model and how to put mathematics behind it. Okay, we have genetic models. We can engineer cells to do or not to do certain things. Okay, and finally, or third, we have model environments. So if I want to be sarcastic, I could say that what wet lab people do, they study model organisms in model environments. So modeling is not alien to the wet lab because mostly all what we do is kind of a modeling. Now we will of course concentrate on the computational models, but not all of them, mostly on modeling things at the community level. Basically what we try to do, we try to go from the genotype to community phenotypes, and this is not a very straightforward thing. Now, there is a last class of models. These are the so-called evolutionary game models, which have a venerable past. Okay, but this is a very specific area where you study let's say, the gene composition of small ecosystems, smaller or larger ecosystems. This is a very well developed field. We are not going into it, but I will give you one example since it was used to. Since it was applied to quorum sensing. But these are the studies where you see the words altruism, kin selection, and the like. Okay, now what we have to know is a very simple thing. And this is and this, fortunately or unfortunately, this remains true. Okay, now let's see a few regulatory models. These are very simple. All of you know this. Okay, so this is a very simple regulatory model of AHL signaling, which relies on an autoinduction loop, upregulation or down, okay, so regulation of the genome, and public goods sharing. So what happens here that we have a very simple regulatory mechanism, basically, which is based on the balance of external and internal signal concentration. This is so simple that it cannot be simpler. So it is a very, very elegant thing. And what I usually tell my students is that in techniques, let's say the doorbell is a similar thing where we have a regulation based on a solenoid. Okay, this is very similar to that. It cannot be made simpler. Of course, in other organisms, we have a bit more players, but the idea is that we have so-called public goods which are secreted, which are sent outside from the cell, and these are no longer the properties of the cell, but these are shared. And these public goods can be then abused, they can be parasitized upon. So here we have this nice social biology terminology, which is based on cheaters, altruism, and so on. Okay, now if we look at the regulatory models, we do not have too many of these, much more than I originally thought. But we have certain repetitive patterns on the chromosomes, and these simple or relatively simple regulatory mechanisms are then modeled in the way Giordano showed us very nicely. Okay, which is basically a kind of a reaction kinetical thing, modeling scheme. Now, in the next step, we have to integrate the signals. Integration of the signals is not a straightforward thing because, let's say, higher organisms use the neural system to integrate the signals. Here, we can believe this or not. Okay, we have the simple, almost Boolean type behaviors of signal connections, and here we come to this. Here I have to mention that the vocabulary, the terminology used by the engineers and by the biologists is very different. Moreover, it varies from lab to lab. For example, these things they work like switches. In technology, this is called a switch, not because it always remains on, just because it has a kind of a bistable behavior. It has a lower and a higher level, okay, and then it might alternate, okay, but it is not a continuous kind of a regulation. Okay. Now, one of the nice things is that in these systems, which are, as we heard now several times during this course, which are much more complicated than this one, but we can produce knockout mutants. So we can knock out signal synthesis, and then we have a mutant which does not communicate, which will follow the order. So it is like a worker in a factory. It follows the orders, it spends its energy and resources, okay, but it cannot give order to others, okay. Now, this is a different type of a mutant where we knock out everything which is downstream of here, which means that this mutant doesn't do anything. It will just consume the resources. And this is a very greedy cheater type. We can say that way. And we can also make a third type of mutants which are good for this modeling studies, where we only, let's say that this is RHL now. So we only knock out one of the secreted goods. Now, what does this mean? This cell will use the entire quorum sensing machinery, only it will not secrete, it will not produce, let's say, RHL or an exoenzyme, okay? So this is a kind of a complex thing that to compare this and this one, okay? Because here, this is greedy because it doesn't turn on anything which is expensive. This is much less greedy because it only doesn't do this, but it does many of the things which may or may not be necessary for cooperation with others. Okay, now let's look at the lab models, which all of you know, but which is important because sometimes it is not obvious from the studies which system the actual data refer to. So liquid cultures can be, from the modeling point of view, they can be characterized as systems where everybody is in connection with everybody else. It's a perfectly mixed medium, which means that when one cell has one neighbor in one moment of time, it will have a completely different neighbors because the system is shaken or mixed. Okay, in this system, everything is global, which means that if you consume the food, the entire food level will decrease. So there is nothing which is confined to a local neighborhood. On agar plates, we have a completely different situation because because here we model an open system, if a front of growth proceeds, and in front of it there is the open space. Okay, so this, at least not on this one because here they already reached the limit. Okay, but for growing colony, this is an unstirred and local communication environment. Finally, we have fermenters when we can vary between the two at will, because there are fermenters where you have a flow through which mimics almost an open environment. Okay, so but these are the three types. Now we come to the model, and I have somewhat deliberately I chose three kinds of models that I would like to speak about. One is the most conventional one, which is not adapted to quorum sensing yet, or hasn't been adapted yet. I will speak about individual agents and hybrid models. Now, what is the difference? In the continuum model, everything is treated as a liquid. So the food is a kind of a liquid that diffuses. Signals, if they were represented, they would be also a kind of a liquid that diffuses. Bacteria are also considered as a continuous material which diffuse. Now, this is the most traditional thing. Now, from individual agents, okay, when we do individual agents, each agent is a bacterial cell. Okay, so we use individual representations for the bacterial cell, and they do something that we tell them to do, and we can look for emergent properties. Emergent means that we give certain rules to the cells, and then look at the ensemble of the cells, and then we will find certain things happening. Okay. Now, in the hybrid models, we have individuals. But we put them into a diffusing environment. Now, why is this important for quorum sensing? Quorum sensing is based on diffusible chemical signals. So you cannot do a very reliable modeling of quorum sensing without allowing some kind of a diffusion. Okay, quorum sensing was explicitly studied in two models, but nevertheless we go through all of them. Now, but let's see the two general goals that we have with modeling. We either model something with a very detailed mathematical description, and we don't really want to know why is it precise, but we want precision. We do this when we have a large fermenter. We don't want to lose the contents because it can be millions of dollars. Okay, so we measure everything and then try to do, usually they do a sophisticated learning algorithm that tell people when is something bad happening. Okay, now in research, so this is an industrial. In research we do something different because we try to make the models as simple as possible so that we could somehow identify what is causing the phenomenon. So this is a minimalistic approach, and the other one is a kind of a brute force many parametric approach. Now, so diffusion only models. This is one of the earlier things that people have done, and this was done long before the advent of quorum sensing, maybe before molecular biology has ever started. So what the kind of the flagship finding of this field is that they try to model colony morphology, and colony morphology is a function of let's say agar concentration and food concentration in the agar plate, and then setting up diffusion-like experiments. Mathematically, these are called reaction diffusion experiments, reaction diffusion equations. You can quite easily reproduce these branched patterns which were previously thought to be mysterious. Okay, now we know that these are much more frequent than the simple patterns. Okay, now we come to one of the models which was already used to quorum sensing, and this is the so-called cellular automaton model. This is again not a completely new invention, but it was only used for quorum sensing, I think, within the last 2 years. So what happens here, that we have a kind of a rectangular grid where in each cell we have an agent, okay? And the agents, they play pairwise game with each other. This doesn't sound very biological, but I'm reproducing it, okay? So they play pairwise games with each other, okay? And in these games, you have to determine who will win. Now, one way or one idea to represent biological bacterium communities, saying that the one which is metabolically more efficient will win with a certain probability. And if you do this for a protracted period of time, game, certain bacteria will win. Okay? Now this has been used to model quorum sensing in a system which is more reminiscent of the gram-positive case. They had 3 genes, so this was the genotype: signal production, signal sensing, and response to signal. And you could choose if these genes were on and off. So 3 genes on and off give you 9 genotypes. And they started this competition, playing pairwise games with many repetitions. Okay, and what they found that sometimes the so-called QS phenotype, in which all 3 genes are on. Signal production is on, response to signal is on, and cooperation, which means secretion of a factor, is also on. Okay, so this phenotype, this is the green phenotype, could win. Let's say could occupy, let's say, half of this imaginary space. But there are, you can read the things there. Okay, so but there are certain questions we have to ask. Okay, so this is not a real space. This is a kind of an imaginary genotype space. Okay, it is drawn as a real space, but it is not a physical space. Okay. Now, these strategies are often used for modeling small ecosystems, and they are thought to, at least people who work in evolutionary game theory, they trust this approach. Okay, here there were 2 interesting findings that were found. First of all, the QS genes were sometimes winning. Okay, but when were they not winning? When you mix the system. Mixing was done by a kind of a reshuffling rule. So imagine that you mix the system after every step of this pairwise game. So now, what happens is that the cells will never have the same neighbor. Okay, and under these conditions, QS doesn't survive for them. So it shows that there is some kind of a link between the mixing of the system and the viability of the QS phenotype. But in any case, what they found was that the 3 genes survive together, okay? Which means that you have cooperation only with communication. And this is a kind of a likely scenario. So this is a very famous model. This is called the Reynolds Model. It's okay, it's okay. It's called the Reynolds Model, which prescribes very few rules for individuals within a crowd. And this is one, that they should separate. They shouldn't bump into each other, okay? And the other, that they should align their course. These are the first 2, okay? Now, as we see, this is what is happening here. However, there is also something else which is shown here. So cohesion. What does cohesion mean? That so these 2 rules, they show that somehow the agents or the individuals should communicate with each other. Okay, and this one is actually a cheating rule because it says that they should know something about where to go. So this is not true for a crowd of individuals. For example, it says that they should know where the center of the flock is and they should go there. They should try to go there. So as I said, this is not true, but you can modify this rule. For example, you can give a kind of an external direction for the community, and then they will behave like migratory birds going. Now, but this is quite complex, because as we say, they don't want to, the fish don't want to go near the stork, okay? So they don't like the sharks either, okay? And very interestingly, as you see, the shark is over there, and something else is happening at the other side of the flock. Now, these models, which are based on this was then used by various groups many times in various fields, mostly for animal flocks, but this is actually a physical model, because we are modeling the things as physical particles, okay? And they just do something which is a function of the entire community. Now, one of the things was the first time that this was used for bacteria. It was quite some time ago. It was Ben. Yes, thank you. Okay. It's okay. Yeah, it was Ben Jacob and associates in, this was a Nature paper, where they used this flock model in a diffused on a diffusible background. The thing that diffused there was the food, okay, and this kind of a hybrid model allowed them to reproduce this branch, so-called fractal shape, and also the same dependence on agar and food concentrations as we saw before. Okay, so here again there was no quorum related thing modeled, and but the idea of using individuals to model bacterial colonies got quite some attention in that year. Now, one class of models which is using this principle is more like a fermentor model, and it was developed at the Technical University of Delft. This is a biofilm model, and what cells do here, the cells are individuals, okay, and they sit in a diffusible environment where you can have food, and if there is food, then they grow, but they are represented as hard spheres. You then start to push each other, then they divide, and then there will be more cells as there is space, and then there is a kind of a relaxation which will spread the system. This is a biofilm growth model. It can give quite interesting patterns, okay? But so basically this is again how it works. So the cells, once they are born, they are there, they remain there, okay? And as we see, the cell density is kind of constant, okay? So if we think about adapting this to quorum sensing, then we should think about what we do about cells, about cell density. Okay, the good idea about this is that this can mimic open and closed environments, same as fermenters can. Okay, now this was adapted not to quorum sensing, but to cooperation situation, basically EPS production. And it was shown that you can have either segregating or mixing populations, or EPS producers and non-producers. Okay, now we go to the approach that we are trying to use. We are trying to use this flock modeling with diffusible signals, and we try to adapt it to quorum sensing bacteria. Now, the interesting story is that when we compare bacteria, this is Howard Berg's lab who did it. This is a kind of a classical video of swarming bacteria and swarming fish. I mean, there is a great deal of similarity, and this takes us, okay, apart from this conscious behavior when the flock is staying away from something, okay? But this takes us back to this to what I said about the physical nature of these models, because this kind of a behavior, as we see here, is due to the rod-like shape of the bacteria. If you take rod-like, I don't know, plastic chips, uniform, and put them onto a tray and shake them, you find this kind of movement. Okay, so this is a kind of a physical thing. The interesting story about it is that it is not a kind of a classical mechanics like physical situation, but a statistical physics situation. Now, this is what we try to work on, and So the phenotype that we want to study is this kind of colony growth. Okay. Now, if we look at this colony growth, this is a bit different from the ones that we saw before. We don't. I can. Okay. This is a branched pattern, but not like the ones we see with swarming bacterial cells. Why this is, we don't know, because these patterns are far too complicated. Okay, but we have genotypes, we have these bacteria mutants that Vittorio's group produced, and then we have the community phenotype, but from the phenotype we did not want to study the colony shape. What we wanted to study was the speed of growth and the growth of the populations. Why? Because we want to study fitness and viability. Okay. Now the growth patterns that we started with were these three. These are the well-known things. Either you have no growth, no swarming. These are swarming plates. You probably know what they are. You have fast growth. This is typical swarming fractal-like branched morphology, and we have constrained growth. And this we have if there is no if the agar is too tight, or if there is too much food. Okay, so this is where we started, and we made a model which was modeling the growth of a simple dendrite of this system. As I said, we only wanted to see the growth of and the speed of advancement. And this is the model. So we put cellular agents, at the beginning of what we call a racetrack. And in this racetrack, we have food. As we start the simulation, the cells start to eat, start to move around randomly, and they move by trial and error towards the areas where there is food. Okay, as they eat, they collect energy, or let's say let's call this energy. And if their energy content is above a certain threshold, they will divide. Okay, but during all this, they also do what a normal quorum sensing cell is doing. They are emitting signals, and if the signal is above a certain threshold, so this is a threshold-based thing, if the system is the signal level is above a certain concentration, they will turn on the production of the public good. Let's say we can call it RHL or an exoenzyme. And when this enzyme reaches a certain level, then they will start swarming. So each cell can be in three different states. Let's put it this way. One which is the planktonic state, and one which is the activated state where it already starts producing the enzyme, and the third is the swarming state. Okay, now during this, as this will happen, so as this happens, the cells move ahead, okay, and we will have a growth in the population, and as well as we will have a kind of a peak-like distribution of the diffusible materials. And this is what I told you before, so now we go further. What happens inside the cell is a simple program. This is very similar to this Ben-Jacob style program, except that when the cells have eaten and they have space and they can survive, so okay, then they perform one of these actions. Okay, so they either behave as resting cells or behave as activated cells. If there is no food, then they stop. We can call, they go back to a kind of a sporulation style things, or they die. We can also do that. Now, the diffusion is mathematically represented. This is just to show you what happens if we calculate diffusion on a plane, because this is a 2D model. This is not important. Okay, and what we see happening is this: the cells start to move around. If we have, let's say, a lot of food, then we have a large population moving around. If we have less food, okay, then we have a smaller population. So this is the fundamental behavior of the system. But we have the advantage that we can count the individual population. So one thing that we see that at the beginning, we only have the planktonic cells. This is the blue thing. And then we have a transient population of activated cells. And after a while, there will be only swarming cells. Okay. Now, if we do the same kind of a plot in the form of cell as a function of cell density, we see that this system behaves as a perfect density switch, which means that at a certain density, from the resting cells we switch over to swarming cells, but there is also a kind of a transient population. Okay, now how does this compare with agar plate experiments? First of all, this is a wild-type mutant. It produces the signal, it responds to it, it produces the factor, let's say RHL, and responds to it, forms this phenotype, and it says that it runs quite fast in the computer. If we delete signal production, then we have a mutant that does not run in itself, but if we add external signal, it will run. Okay, we see the same thing happening here. So this was a kind of a minimum exercise to show that the system fulfills the criteria that we expect from a quorum sensing system. If we delete factor production, so if we we delete the R gene, this cell will never run, okay, and it will never run in the computer either. Okay, and this all looked good. This all looked good, but we noticed that on the computer models there was a kind of a strange situation. Sometime we had populations that started to run and then they stopped, and this did not correspond to any of the phenotype that we expected. Okay, and then Iris Bertani in Vittorio's group did a large study of making pairwise communities for wild-type mutant. This is a Pseudomonas aeruginosa model. Okay, deleting certain genes and making pairwise combinations. In this, so this is a matrix-like arrangement, as you see, and on the diagonal here, we find the behavior of that cell alone, because this is wild-type against wild-type. This is how wild-type behaves. This is how the various less mutant behave. And here we do see a kind of a third unexpected phenotype appearing, because this is where a cell doesn't swarm, no swarming, okay? But we have something which we called collapse, okay? Because it was speed and then stop. First we thought that it is this kind of a thing, but then it turned out that it is not, okay? And this is why we then switched to studying pairwise combinations of cells. As we see, we have a large variety of patterns, and so these patterns do not behave in a very reproducible way. So we kind of simplified the things to this. We have no swarming, swarming, and collapse. Okay? And this is the behavior of the cells on agar plates and in the computer. So what we see here, that we swarming of a wild-type community is, let's say, we have a large population and it is fast. Okay, if we add the so-called non-communicating mutants, this is still a kind of a stable community, but it is smaller and it is slower. Okay. And if we add this greedy mutant, which only consumes the resources, then we have a small start and then complete slowdown, so complete halt. Okay. Now this is a kind of a partial summary of what we could reproduce with this system. First of all, this is a wild-type community. Now we can take a cell from which the synthase gene was deleted. So it is like a perfect solo model. We can call it by other names too.
That's good, Sean.
No, the parents are still alive, so I call it a solo. All the parents' traits. Okay, and we make a streak of signal. This cell will perfectly track this signal track along. So this is a computer signal track, okay? And this is a kind of an interesting finding, even though not completely novel, that it shows that if you have a quorum sensing regulator, this enables your cell to go towards a certain direction, even in the absence of chemotaxis. Now, this is this so-called less stable population, okay, which we see that, so this is a stable population. This is less stable because here only a small part of this population is producing the signals, okay? And the cells, and transiently die out at certain parts of the thing. So if we look at this, we see a kind of a whirlwind of transient populations. While here, when we have the non-communicating cells, we have a sudden collapse, and this is even nicer. By the way, Adam Kerényi is now here. Okay, so he made all the modeling studies, and what he did here was to take a few cheater cells, so non-cooperating cells, and let's see when it starts again, and put them in front of a large population of healthy cells. And what we see that once the healthy cells bump into this very small number of cheats, they will collapse the community. Now this is interesting because this scenario is like the emergence of an unwanted mutation in a community, and for this small community it makes a complete collapse. But very fortunately, in vivo the situation was not so bad, because what Iris did here, she started to grow what was this? It was a normal community, and at a certain time she spotted cheaters at the end of some of the dendrites. And what we see here, that these dendrites which were bombarded, they stopped. The others went on. Okay, so this shows that this collapse is local. The collapse does not spread in the community, and based on this, we can say that since this model doesn't know anything else but quorum sensing, okay, that's it. That in this community, the bad mutations do not spread, because they do such a strong collapse locally that they cannot be carried over. So basically, this is the summary of our findings. So with this model, we could reproduce the certain fundamental phenotype of quorum sensing communities, and we found, interestingly, that these non-communicating mutants can make a quite stable colony together with wild-type mutants. Okay, so there are certain types like communication minus mutants. They can make stable colonies, but others make a strong immediate collapse. Now, this kind of a collapse is not novel. All these particle systems are vulnerable to collapse situations. Avalanches are typical collapses. Okay, we don't know when they happen, but when they strike, the entire community goes into another phase. So it is like a physical phase transition. Now, but it seems that this local collapse can actually eliminate the bad mutations. And we also started to meditate where the stability of this system lies in, and we have a certain ideas why this happens. Okay, and this is kind of a summary now of the entire lecture. So basically what I wanted to, the message I wanted to tell you that modeling of communities or modeling of biological systems is perhaps, so the best idea is to do experimental work and modeling hand in hand, because otherwise there are so many technical difficulties with the system, with the computer system or with the biological system, that it is very easy to go to sidetrack. Now we spoke about the cellular automata, which was used for QS, and this hybrid model, which was also used for QS. We should keep in mind that all the models are very highly simplistic. Okay, so and even those which look very complex, they have brutal simplifications somewhere behind the scene, and it's better to know and admit them. Okay. So these are my colleagues who collaborated in this. Iris Bertani here, Adam Kerényi and Sergio Netoteja in the University of Szeged, and Dora Bihari at the Pázmány University of Budapest. And we made, so some of this is already published. Now, a few caveats again regarding modeling. So basically models try to do everything very simple, but we have to make sure that they are not simpler than they should be. Okay, this is Occam's razor by Albert Einstein. And one other thing, that while we do the models, while we study our lab models or our computer models, there is one thing that we have to bear in mind, and this is a picture of René Magritte. Those who were at my computer course, they have seen this already. I don't know if you know why this is not a pipe, because this is a picture.